Assassin's Creed Odyssey lied to you. In the game, the player watches as statesman and leader Pericles is assassinated, but that's not actually how he died, at least as far as we know. No, Pericles, a man who led Athens in war against Sparta and had a hand in the construction of the Parthenon, died from an infectious disease. That infectious disease caused the Plague of Athens, but which disease was it? Despite the name given to this epidemic, the Plague of Athens, it wasn't caused by the disease plague, often called bubonic plague, based on one of its clinical manifestations. So what do we really know about the Plague of Athens, and how can we find out what caused it? The epidemic that caused the Plague of Athens may have spread through the Mediterranean before entering Athens through its port. The disease broke out in Athens in 430 BCE, which was already a pretty dismal time for Athens. The city was under siege by the Spartans, the Peloponnesian War having broken out the year before in 431 BCE. The epidemic would last for several years, and up to between 75,000 to 100,000 people died. This has been estimated to represent approximately 25% of the city's population. The Athenian general Thucydides caught the disease and survived, and then he did us all a big favor and wrote an eyewitness account of the epidemic and the disease's symptoms. He tells us that he wanted to make a record of this so that if the disease ever broke out again, it could be recognized. There's something incredibly nerdy about that, and I respect it. Thucydides described 3 main symptoms associated with the disease: diarrhea, high fever, and a pustular rash. Lovely. He also noted that it was very contagious and lethal, leading to the deaths of whole families. There were too many corpses to bury, so people's bodies would just be left where they died. The disease didn't reach Sparta, but it's thought to have contributed to Athens eventually losing the war between the two city-states in 404 BCE. Based on the symptoms Thucydides described, it has been possible to narrow down which diseases might have caused the epidemic. However, historians and physicians disagree on which disease it really was. There are 2 main contenders for the causative agent of the Plague of Athens: smallpox and typhoid fever. Smallpox is a really bad time, and that's an understatement. The earliest written record we have of it comes from China during the 4th century CE, which is a pretty long time after the Plague of Athens. It's caused by the variola virus, which has 2 main variants. Variola major causes most cases of smallpox and the most deaths from smallpox, and variola minor, also called variola alastrum, causes smallpox with less severe symptoms. It's very tempting to name a video game character variola alastrum, and that would not be the first time I've named a character after a disease. Smallpox is specific to humans and spreads from person to person through coughing and sneezing. It has several stages as the disease progresses after a person is infected. The incubation period can last between 7 and 19 days, during which an infected person isn't experiencing any symptoms and isn't contagious. The first symptoms can include headaches, body aches, high fevers, weakness, fatigue, and sometimes vomiting. Then we get the symptoms that is arguably the most well-known symptom of smallpox: the rash. The smallpox rash is really gross. The rash turns into skin sores, which are then pustules or bumps full of pus, and then those bumps scab over and fall off. Approximately 1 in 3 people infected with smallpox died, and people who survived could be left with permanent scars or go blind. Typhoid fever, sometimes called enteric fever, has some striking differences when compared to smallpox in terms of its causative agent and means of transmission. Typhoid fever is caused by a specific species and strain of the bacterium Salmonella, Salmonella enterica cerebar typhi. Other strains of Salmonella cause paratyphoid fever and salmonellosis, a common type of food poisoning. Salmonella enterica cerebar typhi and typhoid fever can spread through contaminated food and water, particularly uncooked food, and this can be helped along through unwashed hands when the bacteria leaves the body through feces and urine. So, working with food after a trip to the bathroom and leaving without a stop at the sink can result in a really bad time. I think that makes this my most disgusting video so far. Symptoms of typhoid fever can include headaches, stomach pain, chills, muscle aches, high fever, weakness and fatigue, nausea and vomiting, constipation or diarrhea, and importantly for this discussion, a rash. This rash usually appears on the stomach and chest. Typhoid fever can have a lot of complications, including pneumonia, kidney and bladder infections, and inflammation of heart muscle, myocarditis, the lining of the heart and its valves, endocarditis, the pancreas, pancreatitis, and the meninges, meningitis. With that frankly terrifying list of potential complications, it's not terribly surprising that typhoid fever can be fatal if left untreated. So, we've established our two candidates for the causative agent of the Plague of Athens and what they can do to a human body. How can we figure out which disease actually caused the epidemic when both smallpox and typhoid have symptoms described by Thucydides? Another important similarity between these two diseases is that they don't infect bone, so they don't create any changes to a bone's appearance in a way that we can observe and link those changes back to that disease. So, in order to study smallpox and typhoid in the archaeological record, we need ancient DNA. Great, that's the whole point of me making this video. Where do we start? How do we find appropriate samples to test? Archaeologists excavated a mass burial pit in Kerameikos, an ancient cemetery near Athens, during the years 1994 and 1995. This was perfect timing for using a DNA sequencing technology to test for pathogen DNA in ancient human remains. The burial pit dates back to approximately 430 BCE, so it fits the time of the Plague of Athens, and the remains of at least 150 individuals were excavated from the site. From those 150 individuals, 3 teeth were randomly selected for DNA testing. This is a really small sample size given the large number of people buried in the cemetery, but teeth are really good at preserving pathogen DNA in human skeletal remains. When we extract DNA from teeth, we drill into the tooth's pulp chamber. Because the pulp chamber is enclosed within the tooth, it's protected from external sources of contamination that could mess up those DNA results. We do this with a sterilized dental drill bit mounted on a Dremel, and what comes out is bone powder. That bone powder is then dissolved so we can get to the cells in there and then extract the DNA from those cells. This study also included 2 teeth from a private dental office in Athens that served as negative controls. To test the DNA samples for pathogen DNA, the scientists used what's called targeted sequencing. During the lab work, before the DNA is actually sequenced and we can see what's in those samples, they added something called primers to the samples. Primers are manufactured pieces of DNA that are designed to match up with the fragments of DNA in the samples. This happens because in order to make DNA, especially ancient DNA, easier to study, we have to make copies of it. Polymerase chain reaction, PCR, the process of making those copies, involves taking that double helix structure that we all know and love and pulling it apart. That leaves us with 2 separate strands of DNA. And what we're adding to the samples is DNA that will match up with those single strands and some little chemical buddies to stitch all of that back together into double-stranded DNA again. Boom. You had 1 double helix, and now you have 2. Do that again several more times. There are way better explanations of PCR living out there on the internet with some really great animations to visualize this process, but let's go back to those primers, those manufactured pieces of DNA we can add to our samples. How do those work when we're looking for pathogen DNA in samples that came from human remains? Primers can be designed to match up with specific genes. That's why it's called targeted sequencing. We're using those primers to target those specific genes. If those genes are hanging out in the DNA samples, the primers will match up with them while we're copying the DNA, and we'll be able to see that the gene is present when we sequence the DNA. If we're trying to find pathogen DNA in samples from a human, we can target a gene that is unique to that pathogen. I will note here that this method is a little outdated now, but I do still see it used in ancient pathogen DNA studies every now and then. So, which pathogens were targeted in this Plague of Athens study? The scientists put together a list of 7 diseases to test for, although smallpox didn't make it on their list. It's not clear, at least not to me, why that's the case. Typhoid was on their list, along with plague, Yersinia pestis, anthrax, Bacillus anthracis, cowpox virus, tuberculosis, Mycobacterium tuberculosis, cat scratch disease, Bartonella henselae, and typhus, which isn't the same thing as typhoid and is caused by the bacterium Rickettsia provosecki. Targeted sequencing for 6 out of these 7 diseases failed. Their DNA wasn't detected in the samples from the 3 teeth from the mass grave at Kerameikos. Instead, all 3 teeth tested positive for the presence of the same bacterium, which was Salmonella enterica cerebrotiphi, the causative agent of typhoid fever. So, what conclusions can we draw from these results? It looks like typhoid fever was circulating through the population of Athens during the disease outbreak that came to be known as the Plague of Athens. Did it cause the Plague of Athens? That's entirely possible. The DNA evidence for typhoid is consistent with the account from Thucydides. Typhoid causes the symptoms he described. However, this is only one study, and the sample size, only 3 teeth, is really small. It's pretty normal to have small sample sizes in studies of ancient pathogen DNA, but in the article about this study, it isn't really explained why only 3 teeth were selected when the burial site had included the remains of at least 150 individuals. If other studies based on the Kerameikos burial site or other burials associated with the Plague of Athens have been published since the study, I haven't found them, at least as of the time of me writing and recording this script. When we look at other epidemics, such as the Black Death in Europe, we have tons of DNA studies looking at which disease caused that epidemic. No joke, I've spent 4 years reading them, and new plague studies keep getting published all the time. These DNA tests looking for plague from this time period have been replicated multiple times because we have a lot of mass graves throughout Europe dating to this time period. We have loads of evidence supporting the disease plague as the causative agent of the Black Death. So is this possible for the Plague of Athens? We would need more sources of DNA first. Ideally, we would need another mass grave from Athens that can be dated to the right time period. In their article, the authors explain that mass burials like the one at Kerameikos weren't really common for ancient Greeks. It wasn't a usual way for them to deal with corpses. So first we'd have to find an appropriate archaeological site, excavate it, and carry out DNA testing. All of that involves getting the appropriate permissions to do all of this work, as well as funding to pay for it. Could you pay me to do the DNA work? Please do. This is my very particular set of skills. So that's what we have for a possible cause for the Plague of Athens. I hope you found that interesting, and who knows, maybe someday we'll have some more ancient DNA studies that explore this. Thank you for watching. Assassin's Creed Odyssey drove me absolutely bonkers because I got burnt out on the open world, but it did something really interesting with the Plague of Athens. You can encounter the disease in the game's tutorial zone, the island of Kefalonia. It's at the core of a quest called Blood Fever, which is metal. This quest uses the spread of the disease as a way of introducing the player to the game's roleplay mechanic. If you choose to spare the lives of some people carrying the disease will spread to the rest of the island, and it can cause some environmental changes. Whatever you choose doesn't really have an impact on gameplay, but I think it's an interesting way of incorporating disease, a historical epidemic, and epidemiology not just into a video game, but into the game's tutorial.